sir. Sir, I will give you a small introduction, then we'll start.
Okay,
sir.
okay.
Yes, sir. Yes,
Okay.
sir. Yes, sir. So, a warm good morning to all. Myself, Dr. Ravi Verma, Associate Professor, Ayurved Mahavidyalaya, Hubli. I welcome you all to the GARBHA SAPATAH 2021 Webinar Series 3, Lecture 5. It gives me an immense pleasure to introduce a very kind-hearted, proficient anatomist, Dr. KAMESHWER Singh sir. I welcome you, sir. Sir is presently working as Professor and Head, Department of PG Studies in Sharir, Institute of Medical Sciences, Banaras Hindu University, Varanasi. Sir has completed BMS, MD, PhD in Sharir from IMS BHU, Varanasi. Sir has a teaching experience of more than 23 years for undergraduate and more than 19 years for postgraduates. Till to date, Sir has supervised more than 12 MD scholars and 10 PhD scholars. Sir has published more than 75 articles in various national and international journals. Sir has presented more than 80 papers in various national and international seminars. Sir has attended more than 80 international and national seminars. Sir has also delivered more than 25 guest lectures in various national and international seminars. Sir was an organizing secretary for a national seminar which was organized in 2017 April. Coming to the achievements of the Sir, Sir has received Sharir Shrestha Sushruta Award from Ayurveda Anusandhan Sansthan, Chhattisgarh. Sir has also received Priya Sikshak Sammana in 2019 awarded by National Medicos Organization. Sir has received Best PhD Thesis Award Agnirasa Seva Sansthan from Mathura. Sir has also received Good International Scientist Award and Lifetime Achievement Award in September 2020 in Trichy. Sir holds many positions in scientific bodies and editorial boards. Till to date, Sir has published one book on Shrotas, the title and important analytical structure in Ayurvedic text, and second book is under publication. Sir holds many membership. Life member of Anatomical Society of India, member of Academic Council of Banaras Hindu University, member of PPC PG Medical Board IMS BHU, life member of Vishwa Ayurved Parishad, life member of National Association of Saudhya Harana, vice president of PG Ayurveda Association Varanasi branch, chairman of DRC DPC of Department of Rashna Sharir, Faculty of Ayurveda, Institute of Medical Science, Banaras Hindu University. With this brief introduction, now I hand over the session to Sir to give a lecture. Thank you so much, sir.
Thank you, Dr. Ravi
Thank you, sir.
Verma ji.
Yes, sir.
And thanks to the organizing committee of Mahavidyalaya Hubli, Ayurvedic Mahavidyalaya Hubli, for giving the opportunity to me for sharing my views regarding to the Garbh poshan and Apara nirman and Nabhi nadi nirman. So this is a very important topic for a development of a individual, and the development of the individual is depend on to the nutrition, proper nutrition, and developing individual and during its development period totally depend on to the mother, because it developing individual during the embryonic period or fetal period not have its own source of the nutrition, so it receive the nutrition from the mother, totally depend on to the mother. And regarding this, our Prime Minister launch mission, National Nutritional Mission, and giving the tagline, your nutrition nation's pride. Likewise, so means nutrition is become very important, for the development of the nation. And each and everybody is a unit of this nation. That's why there is need to proper development of everybody. That's why in our text, the process of the nutrition is described by our different acharyas, Acharya Sushruta, Acharya Charak, Acharya Vagbhata, and both Vritrai and Lagutrai. In all text, process of the nutrition was described in very well manner. And I will share some quotations regarding that, and also I will share the modern point of view of the nutrition, poshan, as well as the structures which are involved in the nutrition. Likewise, the placenta, yani ki Apara, and umbilical cord, Nabhi nadi. Because this topic is given to me to share with you. Are you? I am audible properly?
Yes, sir. You're audible, sir.
Okay, okay, sir. Thank you. And this is given the aim of the nutrition. Why the nutrition should be the proper? The nutrition is proper given in this way. Evam jata rupvanta satvanta sirayucha bhavantanasya moktar satputra putrine hita. यानी कि जो progeny है, the progeny should be the good, should be the healthy, and that's why that progeny can fulfill his and her liabilities towards to the our society, and our society have the different persons. Likewise, the Rishi, Dev, Rishi, and Pita, Mata. Likewise, so they have towards them. Everybody has some liabilities. For fulfilling these liabilities, such type of the progeny should be developed. That's why this aim is described by Acharya Sushruta. Evam jata rupvanta sattvanta sirayucha bhavantanasya moktar satputra putrine hita. And furthermore, I want to share the definition of the embryology. From a single cell to a baby in 9 months, a developmental process that represents an amazing integration of the increasingly complex phenomena. The study of these phenomena is called the embryology. And the field includes investigations of the molecular, cellular, and structural factors contributing to the formation of an organism. So that's why development is start from the molecular level to the structural level. And the molecular level decide the fate of a person, fate of an individual, how he can develop, and further proliferation of the cells, proper proliferation, proper cell death, proper differentiation of the cells is also play the role, and regulate by the our genes. Okay, regulate this proliferation and the differentiation is regulated by the genes, and finally we get the structures, and the structure should be the normal. So in its way, we can see in all three scenarios, and out of we have to study, we can study also the Apara and placenta as well as the umbilical cord. They are properly formed or not formed. We will see later on the different structures of the placenta as well as the umbilical cord. Further, in our text, Acharya Sushruta described the definition of the Garbh as well as the definition of the Sharir. So Shukra Shonit Garbha says the Antakarti Garbh Samudram Garbh Tam Chetna Vastitam Vayu Vijati Tej Janam Pasti Apakilanati. Likewise that, and further Evam Vivardita Sayavada Yada Haspajiva Kran Karnitam Badi Hirangi Rupe Tastata Serinti Sangya Lapte Touch Shadangam Sadaka Chetastam Madhyam Panjam System Savivi. That's mean initially molecular factors according to Ayurveda are participate in the formation of a Garbh. Likewise the Aat Prakriti or Vikar, 25 elements takes part in the development of an individual along with the Suprashonit and Atma. So these are the factors, and this process takes place within the uterus. And 5 Mahabhut, also Akash, Vayu, Nijal, Prithvi, play their role in the proliferation of the cells as well as some enzymatic and some hormonal factors as well as providing some fluids and providing the weight, and also providing some cavity, make some cavity within the body, which are important for the development. So in this way, a whole body is formed along with the normal structures, and this whole body is divided into 6 parts for the study point of view. So this is established by the definition given by the Acharya Susmita. And this whole process from the Garbh to the Sharir, this whole process, process of the development is depend on to the proper nutrition. And our acharyas describe the nutrition. Charak Bhagavata describe in different way. Likewise, गर्भस्य खलु रस निमित्त मारुता धात्म निमित्त च परिवद भरती. तो रस जो है वो वृद्धि के लिए रस आवश्यक है. वैसे ही नेक्स्ट चरक बताते हैं, सुसु सर ने बताया यहाँ पर चरक बताते हैं कि स्त्रिया ह्यापन्न गर्भाया स्त्रीधा रस प्रतिबद्धते स्वे शरीर पुष्टे स्तनान्य गर्भोदय. यानी कि जो रस है, आहार रस है, वो 3 पार्ट में डिवाइड हो जाता है. खुद के मतलब न्यूट्रिशन फॉर द बॉडी ऑफ द मदर एज वेल एज टू द डेवलपमेंट ऑफ द ब्रेस्ट एज वेल एज द डेवलपमेंट ऑफ द फीटस. तो उसके 3 जो पर्पज हैं वो यहाँ पे डिस्क्राइब किए गए. फर्दर मातु चहार रसे कुक्षो विवृद्धते, और मातुर आहार से जो है कुक्षी में गर्भ की वृद्धि होती है. ऐसे वहां पे डिस्क्राइब किया गया. फर्दर माता दीना चरक शारीर में डिस्क्राइब है. माता दीना खल्लु गर्भ कराना भावना संपदस्तता प्रत्यक्ष सौष्ठवान मात्रत चेवो स्नेह स्वेदोभ्य काल परिणामात स्वभाव संसिदे च कुक्षो वृद्धि प्राप्त होती. यानी कि स्वभाव से ही जो है हेल्दी मदर के द्वारा स्वभाव से ही जो है बाय द प्रोसेस ऑफ द स्नेहन ऑन और स्वेदन ड्यू टू द टाइम फैक्टर, ठीक है, कुक्षी में स्वभाव से ही गर्भ की वृद्धि होती है. फर्दर कह रहे हैं कि गर्भस्य नाभि नाट्य, तो नाड़ी रसभा स्त्री है. जो स्त्री है यानी कि माता की जो नाड़ी है उसके साथ गर्भ की नाभि नाड़ी जो है संलग्न होती है. 3 गर्भस्य वृद्धि भवति, और वहां से उसको न्यूट्रिशन मिलता है. दैट इज कॉल्ड द वृद्धिर्भवति, गर्भस्य वृद्धिर्भवति, गर्भ की उसके कारण वृद्धि होती. फर्दर कह रहे हैं चरक शारीर में बताया कात्सर्य शरीर वृद्धि करास्त्री में भावा भवंती तथा काल योग स्वभाव सिद्धि आहार सौष्ठवम अविदात चेती. यानी कि आहार से जो है वो जो मदर, द बॉडी ऑफ द मदर receive the nutrition, and that nutrition also provide received by the fetus. तो स्वभाव से ही ये जो है दोनों का, क्योंकि आपस में संधान नाभि नाड़ी के द्वारा संबंध होता है, तो उसके कारण वो उसको न्यूट्रिशन मिल जाता है. एंड ओवरऑल saying that literature of the Ayurveda clearly says that the ras is most necessary for the Garbh origin as well as proper growth and maintenance of गर्भ. तो रस एक इम्पोर्टेंट फैक्टर है आहार जो कि रिसीव होता है आहार रस से, और रस धातु उसके बाद फर्दर जो धातु हैं उनका पोषण करती है. और इस तरह से रस रक्ति मज्जा शुक्र सभी धातुओं का पोषण होने के उपरांत जो है डिफरेंट स्ट्रक्चर्स आर फॉर्म इन द बॉडी, एंड दिस प्रोसेस इज कंटिन्यूस अप टू द proper development of a fetus from a single cell to a baby in 9 months. Then further Charak Sharir is also described, and Kashyap Samhita Sukhthan में भी describe किया गया है. सेम प्रोसेस है कि 3 पार्ट में वो डिवाइड हो जाता है रस जो है. एक तो माता की वृद्धि के लिए, माता की शरीर की वृद्धि के लिए, दूसरा स्तन्य के लिए यानी कि ब्रेस्ट डेवलपमेंट के लिए, और थर्ड वन फॉर द डेवलपमेंट ऑफ ए गर्भ. मातृपुष्ट अर्थ मेकांश, द्वितीय गर्भपुष्ट, तृतीय स्तन पुष्ट, नार्य गर्भस्थ पुष्यति. यानी कि तीनों के लिए आवश्यक है. आगे कह रहे हैं कि चरक एंड काश्यप हैज गिवन द स्टेटमेंट दैट रस derived from the maternal diet performs these functions: nourishment of the mother, nourishment to the fetus, and formation of the breast milk. So that is very clear. These three functions are performed by the RAS. Here, RAS dhatu of the pregnant woman, nourishment of the fetus, formation of the sunny, and nourishment of the own body. So that is clarified. आगे प्रोसेस बताया जा रहा है कि प्रोसेस ऑफ द फीटल नरिशमेंट है डिस्क्राइब्ड इन द आयुर्वेदा. तो उसमें भी वही बात है कि नाभ्य है से नाड़ी प्रसक्ता नाड्य चापरा अपरा चास्य मातु प्रसक्ता हृदय मातु हृदय दस्यताम. यानी कि चरक डिस्क्राइब दैट इनिशियली बिफोर डिफरेंशिएशन ऑफ द बॉडी पार्ट्स fetus gets its nutrition by upsne and by the process of the upswed. Later on, when body parts are conspicuous, it gets its same nutrition by the upsne permeating through pores of the skin situated in root of the hairs and some part through the umbilical cord. Later on, the umbilical cord of the fetus is direct attachment to the placenta and placenta to the mother's heart. The mother's heart supplies RAS to the placenta. यानी कि कार्डिक आउटपुट ऑफ द मदर that reach into the uterus and uterine vessels, uterine artery provide nutrition to the lacunae, into the lacunae, and from the lacunae fetus receive its nutrition. So this process is also described in Ayurveda. The diet of mother must contain all the RAS derived from the mother's diet is capable to give strength, bulk and complexion. यानी कि जो कलर है एवं ज्यादा रूपवंत सत्वंत सिरवसे भवंत से मुक्ता सत्पुत्र पुत्र नहीं था, उस तरह का जो फीटस के डेवलपमेंट के लिए बल और वर्ण यानी कि सत्वंत बनाने के लिए वो सब कैपेबल है. रस जो है दैट इज कैपेबल टू मेक द फीटस proper development, like along with the bulk and worm. Again, Charak said the fetus has no feeling of hunger and thus has no independent existence. Means totally depend on the mother. Fetus gets its nourishment from breast supplied by the mother only when the 6 components of the conceptions are in normalcy and mother use appropriate diet along with mode of life. The rest is transferred to fetus from mother by the process of upsne and upswed. ये जो दो प्रोसेस हैं उपस्नेह और उपस्वेद initially is very critical condition of developing embryo because its mitochondria are not matured. That's why it totally depend on to the mother, and mother provide nutrition as well as the in term of the respiration, in term of the excretion, in term of the nutrition, in term of the respiration, likewise, or in term of the immunity. So these whole process, whole requirement of the developing embryo is fulfilled by these process, upsne or upswed. And this basically is the process of the diffusion, and this is the passive process, passive nutritional process, attain proper growth and development. Further, Susuta is saying that nadi nadi satsi maturahara bhati तेनो स्नेह नास्य वृद्धिर भवति असंजात अंग प्रत्यंग विभाग तु गर्भ निषेकात प्रवृत्ति सर्व शरीर अवयवानुसारेण रसवाना त्रिगताना धमनीना मुक्त स्नेहो जीवयति. यानी कि जो माता की नाड़ी है वो गर्भ की नाड़ी से बंधी होती है यहाँ पर, और उसमें आहार रस और वीर्य जो है वो उसमें ट्रांसमिट होता है. उसके कारण उसका जो स्नेह है यानी कि स्नेह का जो प्रोसेस है उसके द्वारा गर्भ की वृद्धि होती है, जिससे कि डिफरेंट ऑर्गन्स फॉर्म होते हैं. और अकॉर्डिंग टू द बॉडी जो रसवाना त्रिगताना धमनी जो है वो उनमें रस का वहन होता है, और वो जो है ट्रायंगुलरली मतलब इन द होल बॉडी दे आर डिस्ट्रीब्यूटेड एंड प्रोवाइड द न्यूट्रिशन टू द बॉडी ऑफ द fetus. यथा स्वभावतः खानी मनालेशु विशेष धमनी ना खानी रसो ये रुक चेदे. जिस प्रकार से मतलब मंडाल में है ना जो कमल का होता है उसका जो स्टॉक होता है उसके अंदर जिस तरह से पोर्स होते हैं, लाइक वैसे ही यहाँ पर पोर्स होते हैं. दे प्रोवाइड न्यूट्रिशन. Susut explains that the fetal Nabhi nadi is attached to the maternal Rasva nadi. This also I am told. Dallan comments on this as the fetus gets nourishment in the same way as a tree situated on the bank of a full pond receives its nourishment. Further, Susu explained that the respiration actively and sleep pattern of a fetus are depend upon mother. Sushruta has provided the concept of the gaseous exchange from mother to fetus and fetus to umbilical cord and placenta. Yeah, further saying that the Partantra Garbhasya Nishikat Pravati Garbhasyo Pishneho Seda Vartanam, यानी कि वही प्रोसेस है धमनी के द्वारा मात्र हृदय हार रसो धमनी भी चंदन मानो परामुपेति और लास्ट में कहने की तथा रोम कुपेरु स्नेहो रस एवं च भयो भूतम, यानी कि वही है कि माता की नाभि नाड़ी के द्वारा जो है उसका पोषण होता है, ठीक है, गर्भ का पोषण होता है, अष्टांग संगनी का, तो सेम चीज बता रहे हैं. इस प्रकार से both Vagbhata has given a similar description as that other. However, Vagbhata is in Ashtanga Sangrah opines that from umbilicus rus reaches Pakvasha, digestive system of the fetus. Here rus gets metabolized by the Kayagni of fetus itself and ultimately fetus gets its nourishment. Ashtanga and Bhel Samhita have given same opinion, said the fetus receives nutrition by the Kedarikulya process. So this is the Kedarikulya law of the transmission. According to this theory, the process of the nourishment of tissue can be linked to irrigation of the different fields by water from a canal. यानी कि एक चैनल से न्यूट्रिशन आएगा वो डिफरेंट चैनल में ट्रांसमिट होगा और वहां से जो नियर मोस्ट जो सेल्स हैं that they receive the nutrition. So each we know that the each cell have the lies along with the capillary system and within the capillary transmitting the nutrition and by the process of the diffusion fetus cells receive the nutrition as well as intercellular space that also contain the nutritive fluid, and from that nutritive fluid nutrition takes place by the cells of the fetus, developing fetus. As per the description of all ancient acharya, they had clear concept of fetal nourishment. They divided fetal nourishment into two stages. पहला कि after fertilization till the formation of umbilical cord और after formation of the placenta. दो पार्ट में डिवाइड हुआ, एक तो प्लेसेंटा के बनने तक और दूसरा आफ्टर फॉर्मेशन ऑफ द प्लेसेंटा. प्लेसेंटा के बनने से पहले जो है वही दो प्रोसेस हैं, उप स्नेह और उपस्वेद. इनके द्वारा जो है फीटस का न्यूट्रिशन fetus receive the nutrition. During first stage, two processes are mentioned regarding the nutrition are upsneh and upsved. What is the upsneh? To become wet, become smooth, to conciliate, moisturizing, attracting moisture, exudation or anactus, physiological term of Ayurveda. Likewise, the upsved, the causing to sweat, vapor, moisture, sweat, etc. Process of heating. So this is given regarding to these two terms. Upsneh can be equated with the diffusion. The movement of substance molecules from a region of high concentration to one of the lower concentration is known as the diffusion. और इसको हम ऐसे भी देख सकते हैं कि हाई हाइड्रोस्टेटिक प्रेशर से लोअर हाइड्रोस्टेटिक प्रेशर के तरफ जो है मॉलिक्यूल मूवमेंट करते हैं, बेसिकली माइक्रो मॉलिक्यूल्स. तो दैट्स वाई हाई प्रेशर से लोअर प्रेशर की तरफ जो है मॉलिक्यूल्स थ्रू सेमी परमेबल मेम्ब्रेन जो है ट्रांसमिट होते हैं और इस प्रोसेस में यही होता है. उसके अलावा उपस्वेदन कैन बी इक्वेटेड विद एनी एक्सचेंज विद रिक्वायर्ड एनर्जी. तो इट नीड्स द एनर्जी. और जो पहला प्रोसेस है वो पैसिव है, ये प्रोसेस जो है सेकंड वाला उपस्वेद दैट इज द एक्टिव प्रोसेस. एंडोसाइटिक अपटेक ऑफ maternal nutrients histotroph into the embryo during the organogenesis first trimester is required ATP. तो एनर्जी प्रोवाइड करने के लिए ATP is required and that ATP received by the mitochondria. But initially the mitochondria of the developing embryo is not mature. That's why that stage becomes the more critical, and that's why developing embryos depend totally onto the mother initially, onto the tissue of the mother. Now, by the clarification, it is very clear that by the process of the the nutrients comes to embryo during organogenesis in the first trimester. Endocytic uptake require ATP of the uterine nutrients instead of by the trophoblastic cells, and diffusion of these nutrients from trophoblast to embryonic tissue during hysterotrophic nutrition in the first trimester can be equated with upsne and upse respectively. Now, by the this clarification, it is evident that by the process of the SNE and upse, the nutrients reaches to the embryo during organogenesis in the first trimester. Further, cell membrane, Zona pellucida, Zona strata protects ectopic pregnancy. Corona radiata act as the protecting and the nourishing the ovum, nucleus, and nucleus, germinal vesicle, and germinal spot. Cell substance, plasma, and yolk. Yolk is of two types of formative. Yolk cytoplasm as like the ordinary somatic cells, and nutritive yolk. Cytoplasm is small in amount, consists of the glucose, rounded granules of fatty acids and proteins embedded in the cytoplasm. This yolk is of service in the nourishing the zygote in early stage of its development. Nutrition of the embryo plus inside the, just a minute. Nutrition during the embryonic housed inside the fallopian tube. देखिए न्यूट्रिशन तो fallopian tube से स्टार्ट हो जाता है. fallopian tube का जो फ्लूइड है that provide nutrition to the developing structure. The pre-implantation embryo is housed inside the microenvironment of the oviduct, exposed to and is surrounded by the oviductal secretory fluids and in contact with the oviductal epithelium cells. Provides a stable temperature, optimum pH, and dynamic fluid secretions to support embryo development by producing the factors and nutrients required for embryo cleavage. That are required for the cleavage depend on the just a minute. Oviductal fluid is generated by two compositions: the transduction of the systemic circulation and the active biosynthesis from the secretory epithelial cells of the oviduct. The fluid is composed of albumin, transferrin, glycoproteins, galactose, immunoglobulin, glucose, pyruvate, amino acid, lactate, cytokines, many growth factors, and a monitored gas composition. Effect of the oviductal nutrition on the development of the embryo. The influence of the oviduct on embryo development. During cleavage, the mitochondria of the embryo are immature and do not function. ये बड़ा important process है. During this stage, the main energy supply for the embryo is lactate and glycogen, which are provided by the oviductal fluids and the enzymes required for oxidative metabolism produced by epithelial cells of oviduct. Dr. Verma ji.
सर
हेलो
सर सर
जी,
सर बोल
मतलब अगर कोई ऑब्स्ट्रक्शन वगैरह हो तो बता दीजिएगा.
पक्का पक्का बताऊंगा सर, कुछ भी ऑब्स्ट्रक्शन नहीं है सर. यस सर, यस.
During morula and blastocyst, the mitochondria are finally matured and can use oxygen and glucose to produce their own energy via glycolytic metabolism as they leave the oviduct. Further, how oviduct nutrition protects a developing embryo from the congenital malformation. So, first 8 weeks are very crucial for the development of a developing embryo, because the cells are differentiating in that stage, and during the differentiation of the cells, they become more prone for the teratogens, and teratogens are the factor which can produce the congenital malformations. And that's why due to that cause at the time of the birth, structure can be deformed to this process, this mechanism, this oviductal, you can say nutrition can prevent such type of the scenario. How? Oviductal fluid also contain galactase, superoxide, dismutase, and glutathione peroxidase, albumin, and transferrin, which protects the embryo from reactive oxygen species oxidative stress. Galactase converts ROS into the H2 and O2, SOD1 and SOD2 bind the ROS byproducts and turn them into O2 and hydrogen peroxide. Transferrin acts as a metal chelator to prevent the formation of OH ions from Fe2 plus ions. Albumin is prevent lipid peroxidation. So in this way, different factors play their role to prevent the developing embryo from the congenital malformation. At the same time, oviductal epithelial cells provide embryotrophic factors such as growth factors to promote cleavage and embryo development, EGF, epidermal growth factors, FGF, fibroblast growth factor, IGF, insulin-like growth factor, TGF, transforming growth factors. So different factors play role in the development of the fetus along with the nutrition, because safety is very important, and after that the efficacy. If we produce insulin, we produce any vaccine, so first see the safety measurements. Second, see the efficacy, how much percent that is the efficacy. So that's why to protect developing embryo, first is the protection. First need the protection, safety. Second, as well as because if not survive the developing embryo, why the need of the nutrition? So safety and survival of the developing embryo, developing structure is very much needed, and that's why it can receive the nutrition and get the further growth. The role of oviductal nutrition in development of embryo. Oviductal fluids provides growth factors, provide nutrition, help in the stability of embryo against maternal immune system. क्योंकि जो developing embryo है या you can say जो zygote है, that is a foreign body for the mother, and immune system of the mother respond against to the this structure. That's why some factors also have which can minimize the immune system of mother, and that's why embryo can prevent, protect. Protection of the embryo from oxidative and heat stress. These are the some scenario. And the nutrition and after implantation. After implantation, what will be the scenario? After implantation, two principal pathways have evolved to transfer nutrition from the mother to her fetus. These are termed histotrophic and hemotrophic. एक तो है जो कि सेल से डायरेक्ट न्यूट्रिशन मिल रहा है, दूसरा है कि ब्लड से, फ्रॉम द ब्लड ऑफ द मदर fetus taking the nutrition. तो आफ्टर इम्प्लांटेशन इनिशियल स्टेज में हिस्टोट्रॉफिक टाइप का न्यूट्रिशन मिलता है, और लेटर स्टेज में आफ्टर डेवलपमेंट ऑफ द प्लेसेंटा हेमेटोट्रॉपिक nutrition will be started. First, histotrophic is an extracellular maternal derived from the endometrium and the uterine glands. Uterine glands are filled with the glycogen and the lipids, and during the excretory phase, the glands enlarge in the size as well as they become full of the nutritive substance in the form of the glycogen as well as to the lipid. Uterine glands that accumulates in the space between the maternal and fetal tissue. It is phagocytosed initially by the profectoderm. When implanting the developing embryo, that phagocytose to the, you can say, endometrium, okay, for the implantation, and later by the trophoblast of the placenta or the endoderm of the yolk sac before implantation and formation of the placenta. By contrast, hemotrophic nutrition is the exchange of blood borne material between the maternal and fetal circulation once the placenta is established. तो very clear है कि before development of the placenta, histotrophic type of the nutrition is required and provided, and after development of the placenta, hematotrophic type of the nutrition is provided. Now the pathway of traveling nutrition from mother to embryo during first trimester by direct observation in vivo using a histotrophic revealed it to be a clear liquid with no evidence of the blood discoloration present in the IVS during first trimester, which is histotroph secreting from the uterine glands and present in the human during the first trimester of pregnancy. The glandular secretions are delivered into the IVS. From the IVS, intervillous secretions. From the IVS, the maternal products phagocytosed by the trophoblast, then trophoblast, and then into the villus core, from where they diffuse freely through the extracellular fluid into the exocoelomic and amniotic cavities, and then into the secondary yolk sac. The secondary yolk sac is directly connected to the embryonic gut and possess a rich vascular plexus at an earlier stage of pregnancy than do the placental villi. The pathway of the histotrophic nutrition, histotrophs from uterine glands to enter into the intervillous space taken up by the trophoblast, where they digested and utilized in local synthetic pathway or passed into the villus mesenchymal core, then absorbed by the epithelia of the yolk sac, and then absorbed by the amnion and chorion, passed to embryo through the waterline circulation. Effect of the histotrophic nutrition during embryo development. The first trimester of pregnancy coincides with a major phase of the organogenesis in the human embryo. This is consistent with the measurement in vivo demonstrating that the oxygen tension within the placenta is low during this period, and that metabolism with the fetal and placental tissue is largely anaerobic, and the anaerobic glycolysis to meet their metabolic demands during this phase of development and the accumulations of glycogen within the synthetic trophoblast illustrated here are consistent with this view. Now the factors of metabolic transition in embryo. तो पहला factor है कि different nutrient compositions as the embryo travels through the oviduct and later on implanted into the uterus. Second, maturation of the mitochondria to enable the embryo to establish its own metabolism. So initially, embryo cannot perform the metabolism. That's why ready-made food nutrition is required. Later on, it's the mitochondria of the developing embryo is become mature. That's why it can perform its own metabolism. And third one is a change in oxygen tension parallel to this shift is in metabolism during first trimester. Histotrophic nutrition confer benefits on the embryo by minimizing the risk of damage to DNA or disruption of signaling pathway by reactive oxygen species generated during aerobic metabolism. These species are so reactive that many of their interactions are diffusion limited. Hence, the physiological level of the antioxidants cannot provide complete protection. Limiting production of these species by restricting the oxygen supply and providing nutrients via the histotrophic pathway may provide the optimal environment for correct cell differentiation. So that's why पहला तो सेल ग्रोथ होता है, दूसरा सेल डिफरेंशिएशन होता है, और तीसरा है कि ड्यूरिंग द सेल्स डिफरेंशिएशन सेल्स आर बिकम वेरी प्रोन फॉर द टेटोजन, तो उनसे भी प्रिवेंट करना है, अपने फिजियोलॉजिकल अदर फैक्टर से उससे भी प्रिवेंट करना है. तो नंबर ऑफ फैक्टर्स आर hazardous, may be hazardous for the connection unstable. Now the nutritional process of Garbh as per Ayurveda, as per the modern medical science. तो पहला है कि by the process of the से ये बात हो गई, और मॉडर्न के हिसाब से हिस्टोट्रॉफिक न्यूट्रिशन during the critical period of the pregnancy in first trimester. दूसरा है कि some nutrition through long pupina and major nutrition through the Nabhi nadi by the process of the snake. Modern nutritive component from glandular secretion of endometrium and maternal blood vessels to the blood vessels to tertiary coronary villi of the fetus. Though Nabhi nadi and Apara are important for the providing the nutrition, और यहाँ पे exchange of the nutrition waste products through the placenta barrier. Now the placenta, this is the important structure, feto-maternal structure basically, and most part is participate from the fetal side and developed by the chorion in the form of the primary, secondary, and tertiary villi, and from the maternal side is the blood. Blood is filled in the lacunae, so that's why it the placenta in the humans become the hemochorial type. The type of the placenta become the hemochorial, hemo from the side of the mother, chorion from the side of the fetus, and it is a basically 4-layer membrane, and basically it is a membrane. It provides the surface area and also provide the, it acts like a barrier and like at a semi-permeable membrane, and by the process of the diffusion, high from the high pressure to low pressure, the ions nutrition, you can say micronutrients can exchange from one side to another side. So main function of the placenta is the respiration plus excretion plus you can say provide the nutrition and also provide the immunity from the maternal IgG. It can provide the immunity to the developing fetus. Immunity is very much important, as well as all factors are very important. And first need to the survival, then the nutrition, and then the prevention from the pathological factors. So these all functions initially performed by the placenta. That's why it becomes a very important structure. In Ayurveda, it is also called the Jarayu. Satpat Brahman has mentioned that the Garbh is covered with ulva, Varnius caseosa, and Jarayu, amnion, and the Korean both together. Jarayu is above the ulva. This protect the embryo. Formation of the Apara. According to Susut has described the process of the formation of Apara. He opines that after impregnation, the opening of the Artha Shrotas are obstructed by conceptions. Thus, the Artha menstruation is not visible after conception. This Artha being repelled from downward passage goes upward and gets accumulated, which is known as the Apara or Jarayu. So basically, Artha after you can say फर्टिलाइजेशन ऑफ द ओवम या गर्भ की उत्पत्ति के पश्चात जो है जो आर्तव है उसका विजुअलिटी वाज बिकम ऑब्स्ट्रक्टेड या आर्तव कैन नॉट बी सीन इन द मदर्स एंड दिस आर्तव इज डिवाइडेड इनटू 2 to 3 parts. First part for the development of the placenta, second part for the development of the breast, and also it also provides the hormonal scenario for the developing structures. So as well as placenta play the role in the also secretion of the some hormones, progesterone, estrogen, and relaxin, etc. So that's why these role which was performed by the placenta also performed by the Artha after get the pregnancy. यहाँ पे आचार्य सुसु saying that the ग्रहित गर्भाना मार्तम वाहना सोतांश वर्धमान निरुत्यन्ते गर्भेन तस्मात् ग्रहित गर्भाना मार्तम न दृश्यते ततस्ततः प्रतिहत मुदर्मागतम परम चोच्यमानं परदेवते शेष सुधर्मागतम पयोधरा प्रतिपद्धते तस्मात् गर्भिण्यपीनो नत्त पयोधरा भवन्ति. तो श्लोक also saying that जो स्रोतस हैं वो अवरुद्ध हो जाते हैं गर्भ के कारण. इस कारण से जो है ग्रहित गर्भाना आर्तवम न दृश्यते. ग्रहित जिस मतलब गर्भिणी ने गर्भ को ग्रहण कर लिया वह उसमें आर्तव नहीं दिखता है. और वो आर्तव नीचे की ओर तदस्तद नीचे और ऊपर की ओर जाकर के जो है डिफरेंट डिफरेंट स्ट्रक्चर को बनाता है. उसमें एक प्लेसेंटा है और डेवलपमेंट ऑफ द मेमोरी ग्लैंड्स. Hindu commentator of the Susi Samhita opines that besides accumulation of artava, diet of mother also play a role in formation of the apara. Bhagavata has followed Susuta's views. With the knowledge of the modern embryology, it is not clear now that menstrual flow plays any role in the formation of the placenta, but there are some clues that indicates the role of the artava in the placental formation. The microscopic appearance of the placenta, there are many lacunar spaces filled up with the blood and are indicated the obstructed menstruation blood. Ancient scholars may be considered artava in the difference of the pregnancy hormones as they play a role in the placental formation. So, but naturally, the hormones takes play in the role in the development of the placenta. एंड ये जो रोल है वो कॉर्पस लूटियम के द्वारा यहाँ पे प्ले किया जाता है मॉडर्न के पॉइंट ऑफ व्यू से. एंड दैट secrete the progesterone and also maintain the endometrium. ठीक है, and also maintain the pregnancy. After fertilization to till birth of the neonates, there is absence of the menstruation. The menstruation contents as like blood, epithelial cells of the endometrium, the secretions of the glands, and storage of glycogen and lipids are involved in the providing nutrition to embryo initially in formation of the placenta later. According to Ayurveda, the Apara is found by seized arthav, which is Updhatu of Ras Dhatu. Arthav can be correlated with the stratum functionalis of endometrium, while the Ras Dhatu of Garbhasha can be correlated with each and every fluid components, extracellular fluids and intracellular fluids present in the stratum basilis of endometrium. In non-gravitated uterus, the artava can be correlated with stratum functionalis of endometrium, which is as menstrual flow, while in gravid uterus, the seized artava flow can be considered as decidua. Now, these are the process of the formation of Apara. Classical literature according Ras Dhatu, then Artha Dhatu of the Ras Dhatu, then seized after conception, and finally form the Apara. According to current knowledge, as per the current knowledge, basilis of endometrium, stratum functionalis, modified endometrium of pregnant woman, and finally form the placenta. Fetal membranes and the placenta. The placenta is the organ that facilitates nutrition, nutrients and gas exchange between the maternal and fetal component. As the fetus begins the 9th week of the development, its demands for the nutrition and other factors increase, causing major changes in the placenta. Foremost among these in an increase in surface area between maternal and fetal components to facilitate exchange. The disposition of fetal membranes is also altered as production of the amniotic fluid increases. So proper position is also required of the developing placenta, and also it can also perform the proper function if its position is normal. Now, how the development of the placenta takes place. So during the development of the embryo, after formation of the zygote divided into 16 cell stage, and 16 cell stage further develop in the form of the blastocyst. And the blastocysts contain the outer cell mass and the inner cell mass. The inner cell mass is responsible for the proper development of the proper embryo, and outer cell mass are responsible for the development of the supporting structures. Supporting structure likewise the placenta and umbilical cord also. So these outer cell mass, that is called the trophoblast. The trophoblast are the initial structure. And further trophoblast form the extraembryonic mesoderm. Okay. And this extraembryonic plus trophoblast, they form the chorion, and chorion is responsible for the formation of the 3 types of the villi: primary, secondary, and tertiary, along with the some endothelial cells as well as to the syncytotrophoblast. Here we can see the scenario of the developing of the bilaminar disc as in the proper embryo, within the proper embryo, and in the extraembryonic structure, we can see the cytotrophoblast and syncytotrophoblast. Because initially trophoblast cells are proliferate and they release their nucleus as well as the cytoplasm. This cytoplasm and again they retain their cell membrane, and then it is called the cytotrophoblast. So this layer is representing to the cytotrophoblast, and this green color is representing to the syncytotrophoblast. And within the syncytotrophoblast, these are the some space that are called the lacunae, and these lacunae is filled with the maternal blood. Here we can see the maternal blood in the red color. This maternal blood filled within the lacunae, and that's why it provides a source of the maternal from the maternal side to the fetal side. And at this stage, the developing embryo receive its nutrition by the process of the diffusion, that is called the upsnehan and upsvedan. And upsnehan is the passive process, upsvedan is the active process. So in this way, and these lacunae also receive the some arteriole and also the some venules. From the arterioles, blood reach into the lacunae, and from the venules, blood return to the in the maternal circulation. Okay. And other way, this embryo developing embryo also developing some by the angioplastic tissue, angioplastic tissue. Okay. Developing the some blood vessels in the within the body of the embryo as well as in the umbilical cord. Here we can see the further scenario of the development of the placenta. And as I have told that the placenta is a 4-layer membrane. The 4-layer membrane are representing in the at the alveoli level, villi level. Okay. Villi are of the 3 types of the villi are formed. First from the primary villi, second from the tertiary villi, third from the second from the secondary villi, and third one is the tertiary villi. So 3 types of the villi are formed, and this diagram representing the all types of the villi. This is the primary villi, this is the secondary villi, and this one is the tertiary villi. Primary villi contain only the in between and middle is the cytotrophoblast, and outermost is the syncytotrophoblast. So these some you can say process are formed, and they contain 2 layers. So this primary villi is the 2-layer membrane, and this one also have the you can say mesoderm core within the cytotrophoblast. That's why it become the secondary villi, and it has the 3-layer. It is called a 3-layer membrane. Outermost is the syncytotrophoblast, then the cytotrophoblast, then the mesodermal core. And furthermore, developing embryo also develop the endothelial cells, yani ki capillary system, and this capillary system is become present within the this core of the mesoderm. That's why this tertiary villi become the 4-layer membrane. 4-layer membrane, outermost is the cytotrophoblast, then cytotrophoblast, then the mesoderm, then the endothelium. So that's why we can see blood is filled in the endothelium, capillary of this villi, and outside of this villi is present the lacunae. Here present the lacunae. So lacunae is filled by the maternal blood, and capillary of the this villi is filled by the fetal blood. So no directly maternal blood cannot come in contact with the fetal blood, as well as fetal blood cannot come with contact with the maternal blood. So they are separated by the 4-layer membrane, and this 4-layer membrane is increase its surface area by the formation of the villi as well as by the formation of the microvilli. So that's why area is increased, likewise the intestine, as in the intestine. So this is the basic concept of the the placenta. Placenta is the 4-layer membrane. 3 types of the villi are formed, and furthermore, each villi form a anchor likewise this. So these villi become the anchoral villi. Okay. Or this is the maternal side, and this is the fetal side, and this one is the maternal side. And these are the lacunae. They are filled by the blood, and this blood is also come within the intervillous space. These are the 2 villus, or they villus, anchoring villus. So they become initial that anchor, or multiple villi are formed, and between the villi present the space, and this space is filled by the maternal blood. And within this endothelial cells, capillary are present that are filled by the fetal blood. Here we can see. This one is the scenario of the villi, placental villi. This is the intervillous space, and these are the uterine artery providing blood to the fetus. So these are the free villi. Here we can see the blood here. Okay, and these are maternal vessels providing blood to the intervillous space, and these are the this is the umbilical vein. This providing the oxygenated blood to the fetus, and these are the two umbilical arteries that receiving the deoxygenated blood from the fetus and returning to the you can say returning to the maternal side. Okay, returning to the maternal side. But directly blood of the fetus cannot be contact with the mother. Only by the process of the diffusion, all the excreta is reach into the maternal blood, and from the maternal blood, oxygen reached by the process of the diffusion reach into the fetal blood. So this is another scenario. Syncytial node. Frequently, the syncytium becomes very thin, and large pieces containing the several nuclei may break off and drop into the intervillous bloodlets. These pieces, known as the syncytial nodes, enter the maternal circulation and usually degenerate without causing any symptoms. Disappearance of syncytial trophoblast cells progress from the smaller to the larger villi, and although some always persist to the large villi, they do not participate in the exchange between the two circulation. And this is the structure of the placenta. So we can see the fetal side as well as the maternal side. This is the fetal side attached with the umbilical cord. This is the maternal side, contain the cotyledons. 15 to 20 cotyledons are seen on the maternal side. And this is the another diagram of the placenta, contain the villi as well as the intervillous space, contain the maternal blood. This is the another structure of the placenta. Here we can see this totally filled by the maternal blood, and this is the umbilical vein, umbilical artery. Okay. Full term placenta, seen likewise this fetal surface, maternal surface shows the cotyledon. Another diagram showing the blood scenario within the placenta and the villi, anchoring villi. Now the circulation of the placenta. Cotyledons receive the blood through 80 to 100 spiral arteries that pierce the decidual plates and enter the intervillous space at more as less regular intervals. Pressure in these arteries forces the blood deep into the intervillous space and breathes the numerous small villi of the villus, 3 oxygenated blood. As the pressure decreases, blood flows back from the chorionic plate towards the decidua, where it enters the endometrial veins. Hence, blood from the intervillous legs drain back into the maternal circulation through the endometrial veins. So ये प्रोसेस होता है प्लेसेंटल मेम्ब्रेन, जैसे कि बताया कि 4 लेयर मेम्ब्रेन है, आउटर मोस्ट इज द साइटोट्रोफोब्लास्ट है, देन साइटोट्रोफोब्लास्ट, देन द मेसोडर्मल लेयर, एक्सट्रामेडिंग मेसोडर्मल लेयर, देन द एंडोथेलियल लेयर। और हीमोकोरियल टाइप का प्लेसेंटा कहलाता है क्योंकि मैटरनल साइड से ब्लड उसमें पार्टिसिपेट करता है, दैट इज द हीमो। और कोरियोल फेटल साइड से कोरियोन, कोरियोन जो है 3 टाइप्स ऑफ द विलाई फॉर्म करता है, प्राइमरी, सेकेंडरी एंड टर्शियरी, दैट्स व्हाई इट इज कॉल्ड द हीमोकोरियल। फंक्शन ऑफ द प्लेसेंटा, पहला तो एक्सचेंज ऑफ द गैसेस, यानी कि ऑक्सीजन प्रोवाइड टू द fetus and from the fetal side carbon dioxide released into the into the maternal blood. Second one is the nutrition, exchange of the nutrition and the electrolytes such as the amino acid, free fatty acids, carbohydrate and vitamins is rapid and increase as the pregnancy advances. और उसके अलावा इम्यूनिटी के लिए IgG, immunoglobulin, तो begins to be transported from a mother to the fetus approximately 14 weeks. In this manner, this fetus at approximately 14 weeks, in this manner, the fetus gain passive immunity against various infection diseases. Newborn begins to produce their own maternal immunological immunoglobulin G, but adult level are not attained until the age of the 3 years. Hormone also produced by the placenta, that is the HCG, gonadotropin, और HCG जो है वो सिंसाइटोट्रोफोब्लास्ट से रिलीज होता है, जो कि सिग्नल देता है डेवलपिंग कॉर्पस लुटियम को, और कॉर्पस लुटियम फिर फर्दर ग्रेट अमाउंट में एस्ट्रोजन प्रोजेस्टेरोन को प्रोड्यूस करता है। एस्ट्रोजन प्रोजेस्टेरोन जो है वो एंडोमेट्रियम को फर्दर डेवलपमेंट या मेंटेन करने में हेल्प करते हैं, एज वेल एज द प्लेसेंटा को डेवलप करने में हेल्प करते हैं। तो इस तरह से एक फीडबैक मैकेनिज्म के द्वारा जो है and they are depend to each other. Also placenta also release the relaxin, that is and SCS is hemochromatotrophine, is responsible for the development of the breast, so that is also developed by the, you can say, placenta. Clinical correlates, placental barrier. Most maternal hormones do not cross the placenta. The hormones that do cross, such as thyroxine, do so only at a slow rate. Some synthetic progestins rapidly cross the placenta and may masculinize female fetuses. Even more dangerous was that the use of the synthetic estrogens, diethylstilbestrol, which easily cross the placenta. So artificial hormone जो है can cross, natural hormone cannot be crossed to the placenta. It is a very you can say natural process, natural scenario. So natural is always best, artificial is not the best. This is for the further description regarding to the villi, primary, secondary and tertiary villi. एक फैक्ट ये है इसमें कि जो एट द टाइम मैच्योरेशन ऑफ द प्रेगनेंसी या एंड ऑफ द प्रेगनेंसी, जो 4 लेयर मेम्ब्रेन है उसकी जो मिडिल 2 लेयर्स हैं वो थोड़ा सा डिमिनिश हो जाते हैं, जिसमें कि आते हैं सिंसाइटोट्रोफोब्लास्ट और मेसोडर्मल कोर जो है, दे बिकम डिमिनिश, दैट्स व्हाई रिमेनिंग ओनली टू द सिंसाइटोट्रोफोब्लास्ट आउटरमोस्ट एंड द इनरमोस्ट एंडोथेलियल, एंड दिस दैट्स व्हाई जो डिफ्यूजन है वो उसका लेवल यहाँ पर बढ़ जाता है। दैट्स व्हाई जो होल डेवलप्ड फीटस है वो दैट नीड मोर न्यूट्रिशन, दैट्स व्हाई मोर diffusion can be done through the placenta by the 2 layer membrane. Initially by the 4 layer membrane, but later on it becomes the 2 layer membrane. Yeah, 4 layer membrane, but 2 layer becomes some dimness. Now the abnormality of the placenta and cord. Discuss abnormalities of the placenta, explains abnormalities related to the length of the cord. तो अम्बिलिकल कॉर्ड की के रिगार्डिंग जो है 2 बातें हैं कि एक तो शॉर्ट कॉर्ड होता है, एक लॉन्ग कॉर्ड होता है, तो उसके बारे में यहाँ पे प्लेसेंटा के बारे में एब्नॉर्मलिटी ऑफ द प्लेसेंटा डिस्क्राइब्स एब्नॉर्मलिटी ऑफ द कॉर्ड इंपेंडिंग ब्लड फ्लो। प्लेसेंटा सक्सिनेटा एक कंडीशन है, 1 or more small lobes of the placenta size of the cotyledons may be placed at varying distances from the placental margins. In case of the absence of the communicating blood vessel, it's called the placenta sporia. Incidence only 3%. and seen likewise this. Then the clinical significance. It is accentuated lobe is retained followed birth of the placenta. It may lead to the postpartum hemorrhage, subinvolution, uterine sepsis, polyp formation. Treatment. Whenever the diagnosis of missing lobe is made, exploration of the uterus and removal of the lobe under general anesthesia is to be done. Now the placental extracorales. Two types of the described, the circumvallate placenta and placenta marginata. So this is the example of the circumvallate placenta morphology. The fetal surface is divided into a central surface shown surrounded by a thickened white ring, which is usually complete. Vessels radiate from the cord insertion as far as the ring and then disappear from view. The peripheral zone outside the ring is thicker and the edge is elevated and rounded. So here we can see the जो edge है वो elevated हो गया है और we can see the cotyledon. So this is the side of the fetal side. The cotyledon is seen on the opposite side. Now there is the increased chance of the abortion, antepartum hemorrhage, FGR baby, and preterm delivery, retained placenta or membranes. This is the placenta marginata. इसमें क्या है कि a thin fibrous ring is present at the margin of the chorionic plate where the fetal vessels appear to terminate. मतलब fetal vessels जो है वो terminate होती हुई दिख रही हैं यहाँ पर margin के पास. Placenta membrana. Placenta जो है एक membrane की तरह develop होता है, मतलब पूरा जो disc like structure है placenta का normal scenario है वो यहाँ पे develop नहीं होता है. Seen likewise this. Clinical significance, encroachment of some part over a lower segment, imperfect separation in the third stage, chance of retained placenta in more, and manual removal becomes difficult. Placenta aggregata and creta and the percreta. These abnormalities are serious variations in which trophoblastic tissues invade the myometrium to varying depth. They are much more likely with placenta previa or with the implantation over a prior uterine incision of proportion. So यहाँ पे we can see the placenta accreta and creta and percreta. So this depend on to the tissue of the placenta, how much approach to the myometrium. So first position is the accreta, normal position condition. This is the normal stage. 75 to 70% and लाई कर रहा है, ठीक है. और यहाँ पे increta 17% it reaching to the crossing to the myometrium, and in this percreta it comes outside of the uterus after crossing to the, you can say, myometrium and the perimetrium. So in this way these conditions become very serious. Likewise this, they are much more likely with the placenta previa with the condition and trophoblastic to invade the myometrium. Placenta accreta, the condition usually, तो तीनों condition हमने बताया. When the placenta is implanted in lower segments or over the previously injured sites as a cesarean section, dilatation and the curettage operation, manual removal, myometrium. Diagnosis. The diagnosis is only made during the attempted manual removal when the plane of cleavage between the placenta and uterine wall cannot be made out. Ultrasound, imaging, color Doppler, MRI have all been valuable in the diagnosis. Then the pathological confirmation, absence of the decidual bacillus, absence of the metabolic fibrinoid layers, and varying degree of the penetration of the villi into the muscles bundles in creta up to the serous layer. Sorry, muscle bundle in creta और उसके बाद creta. The risk includes the hemorrhage, shock, infection, and rarely inversion of the uterus. Cord abnormalities, abnormal length of the cord, short cord. तो या तो cord जो है वो short हो सकता है, long हो सकता है. यहाँ पे cord की बात करें, umbilical cord की, तो umbilical cord एक जो है amniotic जो membrane है उसका एक sac है, उसका एक tube है, और उस tube के अंदर जो है कुछ content लाई करते हैं. उसमें एक main content है umbilical vein and umbilical artery, left umbilical vein and two umbilical arteries, and also fill the water jelly, also fill the allantois. Allantois is small structure from the side of the cloaca, and waterline duct. Also the content of the umbilical cord. So these are the content of the umbilical cord, and basically it is a tube-like structure. And this tube can be the long and short, less than the 20 centimeter in the case of the short, or commonly relative due to an entanglement of the cord around and if it is part. Exceptional circumstances, the cord may be absent and the placenta may be attached to liver as the exomphalus. Likewise, this liver directly attached हो जाता है ना प्लेसेंटा के साथ या प्लेसेंटा लीवर के साथ, तो ये कंडीशन होती है. Clinical significance: prevent descent of the presenting part, especially during labor, separation of normal situated placenta, fever, implantation, acute inversion, fetal growth restriction, intrapartum distress, failure of external version, two-fold risk of the fetal death. Death का जो risk है double fold हो जाता है. Long cord, clinical significance: cord prolapse, cord entanglement around the neck or the body, true knot, false knots, ये सब बन सकते हैं. This is the long cord, very much long cord. Single umbilical artery. In a very rare case, it is found a single umbilical artery, but it can impact onto the development of the fetus, especially to the heart, and different anomalies of the heart, cardiac anomalies can be produced by the presence of the single umbilical artery. Incidence of the single artery is the 0.363% in live-born, 1.92% in the perinatal death, and 3% in the twins. It is more common in twins and in babies born of the diabetic mothers or in the polyhydramnios. So this is an example of the single umbilical artery and umbilical vein. Where is the umbilical artery, umbilical vein? Yes. It is frequently associated with the congenital malformation of the fetus, 10 to 20%. Villi and genital anomaly, trisomy 18 में commonly मिलता है. There is increased chance of the abortion, prematurity, FGR, perinatal mortality. Single umbilical artery यहाँ पे भी देख सकते हैं. Single umbilical artery. Yes. This one. This one artery, this one vein. Cord insertion, Battledore placenta. The cord is attached to the margin of the placenta. If associated with the low implantation of the placenta, there is chance of cord compression in vaginal delivery, leading to fetal anoxia or even death. Otherwise, it has got little clinical significance. एक ही मार्जिन पे अम्बिलिकल कॉर्ड जो है वो अटैच है, जबकि उसको होना चाहिए सेंटर में. It should be on center. Filamentous placenta. The umbilical vessels spread within the membranes at a distance from the placental margin, which they reach is surrounded only by a fold of the embryo. Although their incidence is approximately 1%, filamentous insertion develops in more commonly with placenta previa and multifetal gestations. This is the example of the filamentous placenta. तो उसका जो मेम्ब्रेन है उसी के साथ अटैच हो गया है umbilical cord. Now the vasha previa. If the release of blood vessels happens to traverse through the membranes overlying the internal os in front of the presenting part, the condition is called the vasha previa. And this is the scenario of the normal placenta, and this is the vasha previa. Pressure from the fetal head. Management. In the presence of the fetal bleeding, urgent delivery is essential either vaginally or by the cesarean section. The newborn's hemoglobin is estimated and is necessary. Blood transfusion be carried out. If the baby is dead, vaginal delivery is awaited. Bipartite placenta. Two complete and separate parts are present, each with cord leaving it. The bipartite cord joins short distance from the two parts of the placenta. Tripartite placenta. मतलब तीन उसके जो part हो गए, three lobes are formed. Tripartite placenta is similar but with three distinct parts. Now the cord abnormality is capable of the impeding blood flow, not pulse knots. Pulse knots appear are knobs protruding from the cord surface and are focal redundancies of a vessels or Wharton jelly with no clinical significance. इससे कोई प्रॉब्लम नहीं होता, जो जेली है उसी का मॉडिफिकेशन हो जाता है. Likewise this Wharton jelly is the modification of the Wharton jelly. True knot. Active fetal movements creates cord knotting incidence approximately 1%, and these are more common in the monoamniotic twins. The risk of stillbirth is increased 5 to 10 folds. This is the true knot. So this occurs in the long cord. In case of the long cord loops, the cord frequently becomes coiled around portions of the fetus. Those loops around the neck are termed a knuckle cord. Several large studies have reported one loop of knuckle cord in 20 to 34% of deliveries, two loops in 2.5 to 5%, and three loops in 0.2 to 0.5%. So this is the loop around the neck, knuckle loop. This is called the around the neck, knuckle cord. Liver progresses, contractions may compress the cord vessels and create fetal heart rate decelerations that persist until the contraction ceases. Phonic presentation, cord prolapse or fetal heart rate abnormality is an associated with the liver findings. Phonic presentation may be identified antenatally with the sonography and with color flow Doppler. If present during labor, cesarean delivery is typically indicated. Umbilical cord strictures. This is a focal narrowing of the cord diameter that typically develop in the area of the fetal umbilical inversion. Absence of the Wharton jelly and the stenosis or obliteration of cord vessels at the narrow segment are characteristic pathological features. Most fetuses are stillborn. So this is the umbilical cord strictures. Hematoma associated with the short cord trauma and the entanglement. They may result from a varix ruptures usually of umbilical vein with diffusion of the blood into the cord. May be caused by umbilical vessels venipuncture. यहाँ पे hematoma can be seen very clearly. Now the cysts, true cysts are epithelial lined remnants of the allantois and may coexist with the persistent patent urachus. In contrast, the more common pseudocysts form from local degenerations of Wharton jelly. Now the clinical significance. Single umbilical cord cyst found in the first trimester tend to resolve completely, whereas multiple cysts may portend miscarriage or aneuploidy. Moreover, pseudocysts persisting beyond this can be associated with the structural and chromosomal abnormalities defects, especially trisomy 18 and 30. तो क्रोमोसोमल डिसऑर्डर्स के कारण जो है ये हो सकता है मल्टीपल सिस्ट, umbilical cord cyst. Thrombosis, intrauterine thrombosis of umbilical cord vessels is a rare event. Approximately 70% are venous, 20% are venous and arterial, and 10% are arterial thrombosis. And this is the scenario of the month-wise dietary regimen. And in the Ayurveda, especially in the Charak 10th chapter, Society 10th chapter, dietary regimen is given very beautifully. And if we see initially, first month sweet, soothing and liquid diet, second month sweet, soothing and liquid diet, third month sweet, soothing and liquid diet, and fourth month cooked rice with the curd, pleasant and delightful food mixed with the milk and butter and meat for wild and meat. So in first 3 months, we can see the sweet, soothing and liquid diet. So that indicating that this diet is very safe and and no chance it can provide the source of the tetragen for the developing structure, because up to the 8th week of the development of the embryo, that remain very prone for the interaction with the tetragens, as well as later on the development of the other structures, upper limb, lower limb, as well as the brain is continuous to the whole development, as well as the after month, after the fetal birth. So that's why very planned diet was described in our text, either by the Susud, by the Charak, by the Vagbhat, and by the Harit. So these are the scenario of the diet of month-wise. Now some scenario of the development या nutrition according to Ayurveda and the modern आयुर्वेद का हमने देखा कि मात्र शुक्ल रसवायन नाड़ी गरुणानी पत्ते पत्ता यानी कि उपस्नेहन के द्वारा जो है उसको न्यूट्रिशन प्रोवाइड किया जाता है. मॉडर्न पॉइंट ऑफ व्यू से देखें तो आफ्टर इम्प्लांटेशन 2 principal pathways have evolved to transfer nutrients from the mother to her fetus. These are the terms histotrophic and hemotrophic respectively. Histotrophic is an extracellular material derived from the endometrium and the uterine glands that accumulate in the space between the maternal and fetal tissue. Further, before the formation of the Garbh, nutrition is also important according to Ayurveda. From Ras Dhatu of the parents and from the Sukra and Artav, they combine to form Garbh. तो शुक्र शोणित गर्भा से प्रकृति गाढ़ समुद्रम, तो दोनों का डेवलपमेंट, पहले शुक्र और शोणित उनका भी प्रॉपर डेवलपमेंट होना चाहिए, प्रॉपर फॉर्मेशन होना चाहिए. मॉडर्न में क्या कहते हैं कि सर्टोली एंड फॉलिकुलर सेल्स रिस्पॉन्सिबल फॉर द एक्सटेंडिंग न्यूट्रिशनल and energy support by providing metabolic intermediate or nutrients including carbohydrates, lipids, vitamins, metal ions and amino acids to the development of germ cells, endocrine and paracrine control of gametogenesis. Further, according to after formation of the Garbh till the formation of the Nabhi nadi, Apara, or before differentiation of the body parts by the उपस्नेह और उपस्वेद के द्वारा होता है. ठीक है, मेडिकल साइंस के अकॉर्डिंग after fertilization during the first trimester and from oviductal fluid and histiotrophs of the uterine glands. फिर before formation of the Apara Nabhi nadi by the process of the उपस्नेह उपस्वेद, सेम बात है. और यहाँ पर histotrophic nutrition by the process of the diffusion and phagocytosis. Later on by hemotrophic nutrition after formation of the Nabhi nadi direct from the maternal ras vahi nadi. In this way, from mother's ahar ras after formation of the Nabhi nadi from first trimester through the fetal placental circulation. Finally, this table is explaining the role of रस धातु in the गर्भ पोषण. रस धातु है, दो चीजें बनाएगी, अपरा एजुब धातु इधर स्थायी फॉर्म इन द गर्भाशय. उसके अलावा शुक्र और शोणित ये भी रस धातु से बनेंगे और जो कि निर्माण करेंगे गर्भ का, गर्भ निर्माण. और फाइनली उसी के द्वारा गर्भ की वृद्धि होगी. ठीक है, और यहाँ पे अपरा एजुब धातु बनेगा और उससे फॉर्मेशन ऑफ द अपरा होगा. आर्तव एजुब धातु होगा और उससे अपरा का निर्माण होगा. दूसरी साइड में बिफोर अपरा निर्माण और आफ्टर बाय द प्रोसेस ऑफ द उपस्नेह उपस्वेद. और फाइनली दीज प्रोवाइड न्यूट्रिशन टू द फॉर द डेवलपमेंट ऑफ द गर्भ, यानी कि गर्भ की वृद्धि के लिए. तो कह सकते हैं कि गर्भ का जो न्यूट्रिशन है वो शुक्र शोणित के लेवल से शुरू हो जाता है और उस लेवल पे उनको प्रिवेंट करना. हेल्दी गैमेट्स शुड बी रिक्वायर्ड फॉर द produce the good progeny. That's why good nutrition is required for the gametes, as well as after fertilization also provide the good nutrition, because at initial stage mitochondria is not matured. That's why ready-made energy is required to the developing embryo, and that is provided by the fluid from the uterine glands in the form of the glycogen and the lipids. and later on other uterine fluids also provide, और अकॉर्डिंग टू आयुर्वेदा बाय द उपस्नेह उपस्वेद. उपस्नेह जो है वो एक पैसिव प्रोसेस है, उपस्वेद जो है एक्टिव प्रोसेस है जिसमें कि एटीपी की रिक्वायरमेंट होती है. इस तरह से जो है प्रोसेस होता है. नाउ फाइनली कंक्लूजन. न्यूट्रिशन ऑफ गर्भ फ्रॉम अस्थायी रस धातु ऑफ पेरेंट्स बिफोर द फॉर्मेशन ऑफ द गर्भ, फ्रॉम आर्तव उपधातु ऑफ रस धातु एंड स्त्री बीज after formation of the गर्भ till the formation of Nabhi nadi, Apara, or before differentiation of the body parts. Sanjata Angle. From mother's ahar ras, asthai ras dhatu, after formation of the Nabhi nadi, yeah, Apara. In this, we have seen the process of the nutrition as well as the development of the placenta and the umbilical cord, and the clinical aspect of the placenta as well as the umbilical cord. In this way, this my talk is complete, and thanks to the organizers for sharing my views with the participants, and thank you to the participants for bearing me, and you can say participating in these views. Thank you very much. Hello, Verma ji.
Thank you so much, sir. Thank you so much,
These are
sir.
the references.
Yes, sir. Yes, sir.
Yes.
Sir,
And
with
this
your
one book. One book is written by my PhD students. Pahel Sahi Poshan Desh Roshan. Likewise that.
okay, sir.
जी बोलिएगा.
हाँ सर. सर कुछ क्वेश्चंस है सर. विथ योर काइंड परमिशन शैल आई टेक इट और?
Yeah. I will try my best.
Yes, sir. Please, sir. Sir, there are some questions in YouTube, sir. The first question is, what are the reasons for immatured embryo?
Reason of the immatured embryo.
Embryo, हाँ सर, हाँ सर.
तो जैसा कि पूरा ये टॉक हुआ है, टॉक के अकॉर्डिंग जो है,
हाँ सर,
निकल के आए कि मेन कॉज ऑफ द जो डेवलपमेंट है वो डिपेंड करता है डिफरेंट फैक्टर्स पे. इनिशियल फैक्टर्स आर द शुक्र शोणित यानी कि गैमेट्स. ठीक है. इफ द गैमेट्स आर नॉट प्रॉपरली डेवलप्ड, ओके, प्लस प्रॉपर न्यूट्रिशन, ये और प्लस सम जेनेटिक सिनेरियो, या सम you can say from the before the genetic some chromosomal scenario. So there may be the multifactorial cause, okay, for the development of the immature fetus. And most of the time we have seen the immature delivery की बात हम करते हैं. ठीक है. So immature delivery about the at the month of the 7th जो है वो देखी जाती है. बट आजकल जो टेक्निक आ गई है, आजकल जो टेक्निक्स है कि उसको जो है फीटस को एक इक्विपमेंट है उसके अंदर उसको रखा जा सकता है. इन केस ऑफ द जो ये होता है फिजियोलॉजिकल जॉन्डिस, फोटोथेरेपी में रखा जाता है. तो उसको जो है आजकल आर्टिफिशियली वी कैन प्रोवाइड द एनवायरनमेंट फॉर फर्दर मैच्योरेशन ऑफ द फीटस, है ना.
Yes.
We can provide nutrition externally and provide the environment. Environment is very important because the mother fetus provide the environment, proper safe environment from the external conditions. So initial cause from my point of view of the immaturation are the chromosomal. Most of the, if you see the, if the students will go in the chromosomal scenario, trisomy 18, 13 and other, they can find out the premature cause of the premature birth is the chromosomal disorders. Plus the nutritional, मतलब mother का nutrition और during the pregnancy किस तरह का है, mother का health किस तरह का है, ठीक है. yes. वो भी इफेक्ट कर सकता है. दोनों कॉज जो है कंबाइंड फॉर्म में इफेक्ट कर सकता है.
यस सर. सर और एक क्वेश्चन है सर यूट्यूब में. सर ले लूँ सर?
जी जी.
यस
बिल्कुल श्योर.
हाँ सर. सर व्हाट विल हैपन टू द फीटस व्हेन देयर इज अ कंडीशन ऑफ एक्सेस amniotic fluid in the womb?
या hydroamnios, that condition is called the hydroamnios. ठीक है, and both hydroamnios and hypoamnios, जिसमें कि ठीक है, hydroamnios and when amniotic fluid become less, both conditions are the harmful for the fetus. Okay.
Yes.
So in condition of the hydroamnios, the length of the umbilical cord can be increased, and some, you can say, circles of the umbilical cord maybe become around the neck, likewise that. और इसका कारण ये है कि हाइड्रोमनियोज क्यों हो रहा है? हाइड्रोमनियोज इसलिए हो रहा है कि जो किडनी है, रीजल एजिनेसिस हो सकता है ना?
हाँ,
क्योंकि वहां पर उसका सिक्रीशन प्रॉपर्ली नहीं हो रहा है, एब्जॉर्प्शन प्रॉपर्ली नहीं हो रहा है, इसलिए जो प्रॉपर ट्रांसमिशन ऑफ द एमनियोटिक फ्लूइड है, फॉर्मेशन और प्लस सिक्रीशन, उसमें कहीं ना कहीं ऑब्स्ट्रक्शन है, तो उसका मेन कॉज होता है किडनी। और प्लस इंगल्फिंग, इंगल्फिंग जो है वो फीटल जो इधर यहाँ पे इसोफेगस वगैरह लेवल पे होता है, ठीक है, और ओरल कैविटी से वो प्रॉपर वे में नहीं हो रहा है, वहां पे कोई ऑब्स्ट्रक्शन होता है, इसलिए हाइड्रोमनियोज की कंडीशन होगी, उसके भी फर्दर इम्प्लीकेशंस हो सकता है इन द डेवलपमेंट ऑफ द फीटस। वो स्ट्रक्चर में हो सकते हैं, मतलब लिम्स की डिफॉर्मिटी हो सकती है, कुछ और हो सकता है, जैसे एक तो हाइड्रोमनियोज होता है, दूसरा हाइपोमनियोज। हाइपोमनियोज में बेसिकली लिम्स जो है वो फोल्ड हो जाता है ना, तो फोल्ड होने के कारण लिम्ब की डिफॉर्मिटी मिलती है देखने को। जी,
थैंक यू सर। सर एक लास्ट क्वेश्चन है सर।
यस सर।
Yes, sir. Last question, sir. Sir, what are the chances of getting diabetes to the fetus if the mother is suffering from diabetes? Like अगर मदर को डायबिटीज है तो फीटस को क्या चांसेस है सर, डायबिटीज होने का चांस है या नहीं है सर?
बिल्कुल चांस हो सकता है, उसको बोलते हैं कंजेनाइटल डायबिटीज, है ना?
यस सर।
तो मदर को मोस्ट ऑफ द केसेस में मदर को जो है ये गेस्टेशनल डायबिटीज होता है, हाइपरटेंशन भी होता है ना? उस केस में डिपेंड करता है कि जो फीटस है उसको टाइप 1 या टाइप 2, है ना?
हाँ सर।
दोनों में से कोई भी हो सकता है, लेकिन मोस्ट ऑफ द केसेस में देखिए जो मेटाबॉलिक सिंड्रोम देखने को मिलता है वो कारण होता है मेटाबॉलिक कंजेनिटल मालफॉर्मेशन, और लेटर एज में जाकर के
सर आपका वॉइस चला गया सर। सर आपका वॉइस नहीं आ रहा है सर। सर आपने ऑडियो ऑफ कर दिया है सर। कामेश्वर सर, सर आपने ऑडियो ऑफ कर दिया है सर। हाँ,
अपने आप हो गया सर, मैंने नहीं किया। मैं ये कह रहा हूँ कि गेस्टेशनल डायबिटीज होता है, ठीक है?
यस सर।
तो दैट मे बी द कॉज ऑफ द कंजेनाइटल डायबिटीज।
यस
और कंजेनाइटल डायबिटीज का जरूरी नहीं है कि वो मतलब अर्ली एज में प्रीडोमिनेंट हो, दैट मे बी डोमिनेंट इन द लेटर एज along with the other metabolic disorders। और आजकल जो सिनारियो चल रहा है, लाइफ स्टाइल डिसऑर्डर्स हैं, उसको वो उसमें वो एनहेंस कर जाता है या डोमिनेंट हो जाता है, तो चांसेस होते हैं कि गेस्टेशनल डायबिटीज होगा तो फीटस को भी डायबिटीज होगा। उसके अलावा उसका इंसुलिन सिनारियो क्या है इसके ऊपर डिपेंड करते हैं। टाइप 1 डायबिटीज है ना, जहाँ पे इंसुलिन बनता ही नहीं है।
हाँ।
और टाइप 2 होता है जिसमें कि जो रिसेप्टर हैं वो इनहिबिट हो जाता है ना, रिसीव भी नहीं कर रहे होते हैं। तो चांसेस मेनली जो है टाइप 2 डायबिटीज के ज़्यादा होंगे, टाइप 1 के भी हो सकते हैं क्योंकि वो भी कंजेनाइटल उसमें आ सकता है। मतलब डिपेंड करता है कितना परसेंटेज है, परसेंटेज वाइज डिफरेंट डिफरेंट हो सकता है। आई हैव नो एग्जैक्ट परसेंटेज सिनेरियो।
हाँ सर।
फर्दर you have given asked very nice questions. I will remind this type of the problem and see the literature regarding that.
यस सर, यस सर। सर अभी तो सभी क्वेश्चंस तो हो गए सर।
चांसेस है, मतलब डायबिटीज के चांसेस हैं
है
जी,
सर।
ये कह
हाँ
सकते
सर।
हैं।
हाँ सर। सर नाउ देर आर नो एनी क्वेश्चन सर, सभी डाउट्स तो क्लियर हो गए सर। सो
ओके,
हाँ सर। सर
तो
नाउ
यहाँ
आई,
पे थैंक यू वेरी मच
सर एक सेकेंड सर।
फ्रॉम माय साइड। Okay.
Yes, sir. Thank you so much, sir, for enlightening all the students who were present online and who was watching on YouTube, for enlightening all the knowledge regarding this Garbha poshan, Garbha nadi, everything, sir. So now I request our final year PG scholar, Dr. Krishna Ji Vaidya, to deliver vote of thanks today to our today's eminent guest speaker, Dr. Kameshwar Nath Singh, sir.
Hello.
Krishna Ji, you are audible.
Thanks. Good afternoon to all. Be constituent in your dedication to showing your gratitude to others. Gratitude is a fuel, a medicine, and a spiritual and a emotional nourishment. Deepo bhakshayate, dvam cha prasuyate, yadannam bhakshaye nityam, jayate tadrashi prajaha. The lamp consumes the darkness and produces the dark suit. Similarly, what we eat affects what we offspring. Like that, Garbha Poshana is mainly occurring through the rasa obtained by the mother, what she consumes. As fetus is dependent on the mother, it's mere responsible to care and look forward for her. The health of the mother, mentally and physically, is to be cured by means of the proper nourishment. Nourishment is not just a nutrition. Nourishment is the nutrition in the food, taste, and the aroma, the love and inspiration in the cooking, and the joy of the entire eating experience. So it's a time to say thanks to the person who nourishes by giving a knowledge on the behalf of college and the department. My heart fills with a lots of gratitude and respect for today's eminent speaker, Dr. Kameshwar Nath Singh sir, for not only sparing their invaluable time for us to grace the event, but also enlightening us with their commendable talk on the subject. We really owe you to us. And next, I would like to thank the torchbearers of this event, our beloved HOD of the department, for supporting and inspiring us for enriching the knowledge. Last but not least, this vote of thanks would not incomplete without expressing any gratitude for all the delegates. Thank you, one and all. Thank you, sir.
Thank you, Krishna Ji. Yes, sir.
Thank you very much.
Sir. Thank you so much, sir.
Thank you. Nice to meet you.
Sir, thank you,
Online.
thank you. Thank you, sir. Namaste, sir.